Hello everyone, welcome back to the class. In our previous class, we discussed about the regulation of activity of cyclin-CDK complex. That is, two factors we learned, that is by cyclin and APC-BARC. Cyclin undergoes cycles of synthesis and degradation, and the enzyme, that is ubiquitin ligase, there are two enzymes, that is SCF and APC-BARC. Now we will be knowing the full form of SCF, that is SKP Culin F box. So that is the full form of SCF. So SCF helps in ubiquitination destruction of the G1 phase cycle. So it is having three central components: SKP1 Culin F box protein, ring domain protein. APC-BARC is another regulatory factor which regulates the activity of cyclin-CDK complex, that is Mitosis-promoting complex or cyclosome. It is lacking F box protein. So this image shows the difference between the actions of two ubiquitin ligase, and this will be dealt today. We also discussed the increase or decrease in the level of cyclin will also determine the CDK activity. The CDK activity is determined by two mechanisms, that is phosphorylation or dephosphorylation reaction, binding of CDK inhibitor protein, that is CKI. One, that is protein kinase activity. The protein kinase activity of the cyclin CDK depends on the phosphorylation state of the catalytic CDK subunit. In fission is the M-CDK activity is inhibited by a protein kinase known as V1. Okay, it is a protein kinase. Kinase means it phosphorylates the particular residue. So it is phosphorylating an inhibitory tyrosine residue, and it is inhibiting, or it is making the cyclin CDK complex inactive for a while. Why it is doing? It is checking the premature entry into mitosis resulting into a small cell, that is making that impossible. So it is making sure the cell will attain a particular adequate size for cell division or mitosis. Okay, the opposite for V1 is CDC25. It is a protein phosphate which removes the inhibitory phosphate, which acts as an antagonistic. So V1 and CDC25 as its antagonistic role. If there is a less amount of V1 and more amount of CDC25, it means there is active CDK. If V1 is kinase is there, then the inactivity of CDK cyclin CDK complexes is for sure. So V1 is phosphorylating the inhibitory tyrosine residue of at the 15th position. Make note of this. And V1 is a dual specificity kinase, as it phosphorylates serine or threonine and also tyrosine residue. So we also came across what if there is a loss of function of V1. So there is premature entry into mitosis, small cells are produced, and we also discussed what happens when overproduction of V1 happens. So there is increase in the length of G2 phase, elongated cells are produced. Okay, and we also discussed this chart, the activation of M-CDK. So M-CDK we learned, or we are pronouncing this word M-CDK many times. So today we'll be learning about the role of M-CDK in the cell cycle. So what does it do? What is its main function? Okay, the role of M-CDK. So to begin with, the role of M-CDK, we'll first completely give the abbreviation of M-CDK. Okay, M-CDK. M-CDK is referred to as mitosis cyclin-dependent kinase. It is the M-CDK is present in which stage? That is in M stage. So mitosis cyclin-dependent kinase. Okay, and this M-CDK is also known as MPF. M-CDK is also known as MPF. What is it? MPF? Its full form is mitosis-promoting factor or maturation promoting factor. So what is M-CDK? M-CDK is also called as mitosis promoting factor and also maturation promoting factor. It is making the M-CDK, if there is a presence of M-CDK, it is making the cells mature. It is sending the cell for the next maturation phase. Okay, it is synthesized during this M-CDK is synthesized during S and G2 phase itself, but it is inactive. Okay, when there is a end of G2 and G2 phase and when there is a initiation of M phase, this M-CDK will get activated. So M-CDK complexes, what are their functions? Their main function is they induce chromosome condensation. First, then they break down what? Nuclear envelope. They break down nuclear envelope and they also help in assembly of mitotic spindle apparatus. Okay, and also they have another function, that is alignment of condensed chromosome at the metaphase plate. Alignment, I'll just write a keyword, alignment of metaphase chromosome on the metaphasic plate. Okay, so these are the major functions of M-CDK. So they induce a chromosome condensation, they help in breakdown of nuclear envelope, they help in assembly of mitotic spindle apparatus and alignment of metaphasic or condensed chromosome at the metaphase plate. To perform this activity, so we know that CDKs, what is the function of CDK? So it catalyzes the reaction where phosphorylation happens. Okay, phosphorylation reaction is taking place. So CDK phosphorylates or it catalyzes the phosphorylation process. Okay, the M-CDK function is same. So to perform these activities which has mentioned in previous slide, so these M-CDKs phosphorylate wide range of proteins. So they phosphorylate wide range of proteins. Okay, these proteins are, there are several proteins. One is condensin protein. This condensin protein is very essential for extensive condensation of mitotic chromosome. And M-CDK also phosphorylates lamin protein. This lamin protein, which form a stabilizing network under the nuclear membrane that disassemble during mitosis. Okay, so just consider these two important proteins. So M-CDK phosphorylates condensin protein in order to make extensive condensation of mitotic chromosome, and M-CDK phosphorylates lamin proteins which forms a stabilizing network under the nuclear membrane. Okay, and condensin is a highly conserved 5 subunit protein. So we are now heading towards condensin protein. So this, as I said earlier, so it is helpful in extensive condensation. Okay, it has a highly conserved 5 subunit protein complex. So it is having how many subunit protein complex? 5 subunit protein complex. Among these 5, so there are 2 SMC proteins. Among this complex, so there are 2 major SMC proteins, that are SMC2 and SMC4. So these two plays a fundamental role in structural and functional organization of chromosomes. So these are the key drivers, we can say it. These are the key drivers of the mitotic chromosome condensation. So what did we learn now? Condensation protein is helpful in extensive condensation of mitotic chromosomes, which is phosphorylated by M-CDK. Phosphorylation happens by M-CDK in M phase. There are 5 subunit protein complex in condensation proteins. Among them, 2 SMC proteins are majorly important, that is SMC2 and SMC4. These play a fundamental role in the structural and functional organization of chromosomes, and they are the key drivers of mitotic chromosome condensation. So during mitosis, this condensin protein is phosphorylated, that is insertion of a phosphate group. It is phosphorylated by 3 major kinases, that is cyclin-CDK, polo and aurora B kinase. So condensin is phosphorylated by cyclin-CDK, polo and aurora B kinases in a manner that correlates with changes in its localization, dynamics and supercoiling activity. Now, the M-CDKs also influence the assembly of the mitotic spindle. As I said before, the M-CDK influence the assembly of mitotic spindle. Assembly of mitotic spindle apparatus, okay, that is by phosphorylating proteins, that regulate microtubule behavior. So the protein which acts or which regulates the microtubule behavior, that for that protein the M-CDKs phosphorylate that protein which regulate microtubule behavior. Okay, the net effect of these coordinated phosphorylation, that is by several other kinases families. So by this coordinated phosphorylation reaction, the accurate separation of chromosome during mitosis happens. Okay, M-CDK initiates these changes in microtubule behavior by phosphorylating 2 classes of proteins. So as I said, the phosphorylation happens to the proteins which help or which regulate the assembly of mitotic spindle. There are 2 classes of proteins that control microtubule behavior. This protein includes microtubule dependent motor proteins. I'll take a new sheet. So we are discussing now the M-CDK phosphorylating microtubule dynamic, or the protein which controls microtubule dynamics, that is 2. First one is microtubule dependent motor proteins. Okay, so the M-CDK phosphorylates this protein, and also there is another protein, that is microtubule associated proteins. In short, we'll call this as MAP. Okay, so M-CDK influence the assembly of mitotic spindle by phosphorylating major two proteins that regulate microtubule behavior, which are those microtubule dependent motor proteins and also microtubule associated proteins, that is MAP. After the proper association of all these chromosomes with spindle microtubules has occurred, so there is a association of all chromosomes with the spindle fiber that is successfully done. So M-CDK complex activates, guess who? M-CDK activates APC bar C. So now the cell has got adequate size, and the proper assembly of mitotic spindle has done with the help of phosphorylating proteins that regulate microtubule behavior by M-CDK, and also there is a proper extensive condensation already happened by phosphorylating condensin by 3, that is cyclin-CDK, polo and aurora B kinase. Now that is the complete perfect environment for the cell to undergo mitosis. So now this M-CDK calls for APC bar C to activate that is mitotic events leading up to metaphase. But it also sets the stage for progression into anaphase, that is later. But for now, M-CDK triggers the early mitotic events and also leading up to metaphase. Okay, so first we'll summarize what we learned now. M-CDK phosphorylates many proteins, among them condensin and lamin are the major one. Condensin, which helps in extensive condensation, and the lamin, what does the lamin do? Lamin stabilizes network under the nuclear membrane that disassembles during mitosis, and this condensin have highly conserved 5 subunit protein complex. Among them SMC proteins are important, that is SMC2 and SMC4. They play fundamental role in structural and functional organization of chromosome. And during mitosis, condensin is phosphorylated by cyclin-CDK, polo and aurora B kinase. All these are phosphate protein kinases in which correlates with changes, localization, dynamics and supercoiling activity. After that, M-CDK also helps in assembly of spindle fiber or spindle apparatus, mitotic spindle apparatus, by phosphorylating protein which are regulating the microtubule behavior. So there are two classes of proteins which regulate the microtubule behavior, that is microtubule dependent motor proteins and microtubule associated proteins, that is MAP. After phosphorylating all these class of proteins necessary for necessary for condensation, assembly and stabilization, after that M-CDK activates another major component, that is APC bar C. See here the role of M-CDK. M-CDK is also known as mitosis-promoting factor or maturation-promoting factor MPF. It is synthesized during S phase and G2 phase. M-CDK complex has many functions like induce chromosome condensation, breakdown of nuclear envelope, assembly of mitotic spindle apparatus, alignment of condensed chromosome at metaphase plate. It phosphorylates wide range of proteins. Among them, it phosphorylates lamin protein, which helps in breakdown of nuclear envelope, stabilizes nuke network under nuclear membrane that disassembles during mitosis, and it phosphorylates condensin protein, which induce chromosome condensation. So condensin is highly conserved. As I said, it is a 5 subunit protein complex. Among them, SMC proteins are important. See, SMC2 and SMC4. It has a fundamental role in structural and functional organization of chromosomes. So in mitosis, this condensin protein is phosphorylated by cyclin-CDK, polo and aurora B kinase in an orderly manner that correlates with change in its localization and dynamics, and also supercoiling activity. So M-CDK also influences what? Influences assembly of mitotic spindle by phosphorylating proteins that regulate the microtubule behavior, that is microtubule dependent motor proteins and microtubule associated proteins, that is MAP. M-CDK phosphorylates MAPs, which helps in accurate separation of chromosome during mitosis. So what is the function of microtubule associated protein? It helps in accurate separation of chromosomes during mitosis. Proper association of all chromosomes with the spindle microtubule is also a major factor. Okay, after all this, M-CDK complex activates APC-BARC, which sets a stage for progression to anaphase. So here what does the APC-BARC do? It acts on different substrate at different times. First, its function is in coordination with two homologous mitotic coactivators, that is CDC20 and CDH. Okay, CDC20 homolog 1, that is the full form of it. And thus, the M-CDK does not triggers the early mitotic events leading to the metaphase, but also sets the stage for progression into anaphase we know now. The APC-BARC functions in coordination with two homologous mitotic coactivators as mentioned here, CDC20 and CDH1. Okay, APC-BARC initiates the metaphase anaphase transition through mediating ubiquitination or ubiquitination, we can say ubiquitination or ubiquitination and degradation of M-cyclin, and securin. Okay, so this APC-BARC initiates metaphase anaphase transition through what? By mediating ubiquitination and degradation of M-CDK and securin. So what is the function of this securin? So securin, which holds enzyme separase. It is a ubiquitous cysteine protease. Okay, it is a ubiquitous cysteine protease which is in inactive state. The destruction of securin. So we can say, I'll write it like this: securin is holding separase. Okay, is holding separase and making it inactive at the initial stage. So this destruction of securin allows the separase to cleave SCC1, which allows the separase to cleave SCC1, a subunit of the cohesin, which holds the sister chromatids together. So I'll draw it like this. So securin, so there is securin right here which holds separase, okay, which is in inactive state. So APC-BARC is activated in late M phase by the association of CDC20 or CDH1. The activation of APC-BARC by CDC20 or CDH1 leads to the ubiquitination and destruction of securin. So here I'll write destruction by APC bar C. So what happens? The destruction of securin allows the separase to cleave a subunit of cohesin. So this separase cleaves what? Cleaves subunits of cohesin complex. The pulling force of the mitotic spindle then pull the sister chromatids apart, helping the proper separation or pulling of the sister chromatids apart. So the separase activation is very, very important for breakdown of cohesin protein and also making sure the proper division of the sister chromatids apart to the following daughter cells. Okay. See here, destruction of securin allows separase to cleave SEC1, a subunit of the cohesin which holds sister chromatids together. I'll draw a image here. See, securin, I'll write SEC. Along with that, we have separase SEP, on which APC bar C. No, just APC is not enough. So APC is activated by CDC20. Okay, where this APC bar C ubiquitilates. I'll write the diagram, you will get to know. So this securin is here. The APC bar C adds ubiquitin chain. So now securin is having ubiquitin on it. So it is leaving the separase alone. So this separase is active. This separase was inactive. I'll write it as INA, inactive, because it was bound to securin. When securin left separase became active. So what is the function of this active separase? So consider here there is a chromosome with sister chromatids and there is spindle apparatus attached to it. So this separase help in breakdown of cohesin, I'll write BKD, breakdown of cohesin protein. What happens when there is a breakdown of cohesin protein? It releases sister chromatids. It pulls out the sister chromatids apart, okay, helping the proper segregation of sister chromatids apart. Now you got to know, okay. So here there is APC bar C which is inactive. When CDC20 comes to ubiquitinate, leads to ubiquitination and helps in making that active APC bar C. This active APC bar C will ubiquitinate and degrade the securin. So here securin is there, inactive separase is bound to it. When active APC bar C act on this complex, the securin will be degraded. After that, active separase is free now, it is active, okay, which is leading to cleaving the cleavage of or dissociation of cohesin, leading to anaphase stage. So from metaphase to anaphase, this is the transition what we were discussing till now, okay. There is coherency between two chromosomes or the arms, chromosomal arms. So there was a cohesin which was making sure the pairing or attachment of these chromosomes, right? At the G2 phase it was like this, but after M-CDK's inactivation or M-CDK's activation, M-CDK activation taking place, the chromosome is also undergoing different changes. So here there is cleaved and dissociated cohesin, allowing both sister chromatids to pull apart. So this is the major function of M-CDK. M-CDK. Okay, here M-CDK is influencing the assembly of the mitotic spindle and also helping the separation or segregation of sister chromatids. So there is another important protein which regulates the cyclin-CDK complex. So till now we discussed many aspects which regulated the activity of cyclin-CDK. The regulation, can you just recall? Regulation of activity of cyclin-CDK complex. Which were the factors? That is SCF, that is ubiquitin ligase, E3 ubiquitin ligase, APC bar C, phosphorylation and dephosphorylation action, and we all discussed this. Okay, but we are left out with one aspect, that is binding of CKI. Binding of CKI. What is this CKI? CKI is nothing but a protein, that is CDK inhibitor protein. CDK inhibitor protein. So cyclin-CDK complex can also be regulated by binding of CDK inhibitor proteins. So CKI is binding, so this CKI or CDK inhibitor protein binding with cyclin-CDK complex, it makes or it results into inactivation of cyclin-CDK. So inhibitor is nothing but stopping the functioning of CDK. So this CDK inhibitor protein, that is CKI, will bind to the cyclin-CDK and making the cyclin-CDK complex inactive. So there are two types of CKIs. That is CDK inhibitor proteins, that is CIP bar KIP family, and INK4 family. CIP stands for CDK, I for interacting, P for protein. CIP, CDK interacting protein. The full form of KIP, that is kinase inhibitory protein. The name itself tells everything. CIP, CDK interacting protein, that interacts with CDK, and KIP, that is kinase inhibitory protein, that inhibits, this protein inhibits what? The kinase. Okay, they are, so CIP KIP family includes P21, remember this, P21, P27 and P57. All these are inhibitors. Which belong to which family? CIP KIP family. What is the CIP KIP family? It is a type of CKI. What is CKI? CDK inhibitor protein. What does CKI do? Which inhibits or make the cyclin-CDK complex inactive. Okay, so which are the three major CKIs of CIP KIP family? That is P21, P27 and P57. And we are left with another one major important family, INK4 family, which is a type of CKI itself. So INK, okay, INK full form is inhibitor inhibitor of CDK4, inhibitor of CDK4. Okay, so this family consists P16, P15 and P19. So INK4 CKIs target specifically CDK4 and CDK6. They inhibit their kinase activity by interfering with their association with D-type cyclins. Okay, so this specifically bind to CDK4 and CDK6. See here, CDK inhibitor proteins, that is CKIs, has a role in regulation of cyclin-CDK complex. CKI plus cyclin-CDK complex make cyclin-CDK complex inactive. There are two families, CIP, KIP family and INK4 family. CIP full form is CDK interacting protein, KIP full form is kinase inhibitory proteins. Examples are P21, P27, P57. This inhibits their kinase activities by binding to D, E, A and B cyclin-CDK complex. INK4 family they are the inhibitors of CDK4 and CDK6 as well. Okay, example are P16, P15, P18 and P19. So this inhibits the kinase activity by interfering with their association with D-type cyclins. Here you can see active cyclin-CDK complex is there. P27 is an example for CIP-KIP family of CKI. So when P71 binds with this complex, it makes the cyclin-CDK complex inactive. So that's it for today. In next class, we will be learning about control of DNA replication in S phase. So till now we discussed many aspects from right from cell cycle introduction to role of M-CDK. Kindly revise each and every concept and kindly make a concept map so that you can revise it quickly and you can form a link between each concepts. Thank you.